We're going to move on to chapter 25. I'm going to skip chapter 24. That chapter deals with nutrition, and those of you in the nursing program will be taking an entire course in nutrition, so I'm not sure I can add much to that. If you want to read through that chapter, it's probably a good idea to give you some background. And there is some chemistry in there that's important for organisms, but I don't think you'll need it in your day-to-day professional work. And this chapter is going to wrap up this course. So chapter 25 deals with the urinary system. And we'll start with the kidneys. Kidneys are the major excretory organ that helps get rid of waste that we generate within the body itself. So the digestive system waste was waste that we took into our bodies, didn't use, and then passed through on the other end. The kidneys are going to take care of waste that we generate in the process of metabolism. A lot of nitrogen waste, because breakdown of proteins involves nitrogen, and nitrogen can be very toxic if it's not dealt with. So the kidneys are going to be a major way to do that. And along with that, they are also going to be important for regulating total water volume, total solute concentration in the fluids, regulating ion concentrations in the extracellular fluids. They're very important for maintaining our long-term acid-base balance, excreting metabolic wastes, toxins, and some drugs. And we've already touched on the fact that they produce a hormone called erythropoietin, which is important for regulating red blood cell production in the bone marrow. And they also produce renin, and renin is important for regulating blood pressure. So if you notice, this slide is a little bit wrong. Erythropoietin goes with red blood cells, renin goes with blood pressure. They also activate vitamin D, and they carry out gluconeogenesis if needed. Kidneys are part of the urinary system, and that urinary system, in addition to the kidneys, includes the ureters, which transport urine from the kidneys to the urinary bladder, the urinary bladder, which is a temporary storage reservoir for urine, and the urethra, which transports the urine out of the body. They are shown here in their anatomical positions. Here is a cadaver dissected. So let's take a look at the gross anatomy of the kidneys to begin with. They're retroperitoneal. They're in the superior part of the lumbar region. It's located between T12 and L5. The right kidney is crowded by the liver, so it's a little lower than the left. On top of the kidneys, the adrenal glands sit there, and those are also referred to as the suprarenal glands. They have a convex lateral surface and a concave medial surface, and on that medial surface is the renal hilum, which leads to the internal space, the renal sinuses. The ureters, renal blood vessels, lymphatics, and nerves enter and exit through that hilum, much of the way they did in hilums in other organs we've looked at. So here they are in anatomical position. There are 3 layers of supportive tissue around each kidney. There's a renal fascia that anchors it's an anchoring outer layer of dense fibrous connective tissue. There's a perirenal fat capsule, which serves as a fatty cushion, and a fibrous capsule, which is transparent and prevents the spread of infection to the kidneys. It kind of isolates the kidneys from the rest of the body. Upper parts of both kidneys are protected by the thoracic cage. There is also perirenal fat that provides cushioning. The lower part, though, is susceptible to blunt trauma, especially the right kidney. So a fall, a motor vehicle accident, contact sport injury can work on the lower part of the kidneys. The renal artery is especially vulnerable to injury from a rapid deceleration during car crashes that can lead to lacerations or tears, or just a blood clot within it, a thrombosis. Hematuria is blood in the urine, and that would be an important sign of that kind of trauma, and surgical treatment for that may be required. The internal kidney has three distinct regions: a renal cortex, which is a granular appearing superficial region. The renal medulla is deep to that. It's composed of cone-shaped medullary pyramids or renal pyramids. Broad base of the pyramid faces the cortex. The papilla, which is the tip of the pyramid, points internally. And those renal pyramids are separated by renal columns, which are inward extensions of the cortical tissue. And lobes are the medullary pyramid and its surrounding cortical tissue. And there are about 8 lobes per kidney. So this is another organ with a cortex on the outer part and a medulla on the inner part. There's also the renal pelvis, which is a funnel-shaped tube, and it is continuous with the ureter. There are minor calyces, which are cup-shaped areas that collect urine draining from pyramidal papillae. And there are major calyces that are areas that collect the urine from those minor calyces, and those empty into the renal pelvis, which means that urine will flow from a renal pyramid to a minor calyx to a major calyx to the renal pelvis to the ureter. And here's the internal anatomy of the kidney, figure 25.4, and I suggest you look that over in your text. Pyelitis is an infection of the renal pelvis and calyces. Pyelonephritis is an infection or inflammation of the entire kidney. Infections in females are usually caused by fecal bacteria entering the urinary tract. Severe cases can cause swelling of the kidneys, abscess formation, pus may fill the renal pelvis, and if left untreated, kidney damage may result. But treatment is normally successful using antibiotics. The blood. Kidneys cleanse the blood and adjust its composition so it has a very rich blood supply. The renal arteries deliver about 1/4 of the cardiac output to the kidneys each minute. That's just over a liter per minute. And the arterial flow would be from the renal arteries to segmental arteries to interlobal arteries to accurate arteries at cortical radiate or interlobar. Coming out, the venous flow would be from the cortical radiate, the accurate interlobar and renal veins. So there are no segmental veins. Its nerve supply, the sympathetic fibers from the renal plexus. And again, I suggest you go over these figures in your text. The structural and functional unit of a kidney is a nephron. And there are about 1 million nephrons in each kidney. The nephrons are divided into 2 main parts, the renal corpuscle and the renal tubule. And again, these are the structural and functional units that form urine. These are going to be important to understand when we talk about how we make urine and do what the kidneys need to do. So the corpuscle is made up of the glomerulus. The glomerulus are tufts of capillaries with a fenestrated endothelium. They are highly porous capillaries. They allow for an efficient filtrate formation. And that filtrate is plasma-derived fluid that the renal tubules are going to process to form urine. So the first step in making urine is to get the stuff, the liquid, out of the blood. And that happens here in the glomerulus, and that liquid is called filtrate. That glomerulus is surrounded by a capsule called the glomerular capsule, or traditionally known as Bowman's capsule, after anatomists who described them. It is a cup-shaped hollow structure surrounding the glomerulus, and it's got 2 layers, a parietal layer and a visceral layer. The parietal layer is simple squamous epithelium. The visceral layer is going to cling to that glomerular capillary and has branching epithelial podocytes or foot-like projections. The extensions terminate in what is known as a foot process that clings to the basement membrane, and there are also filtration slits between the foot process that allows filtrate to go into the capsular space. So that filtrate that is coming out of the capillary, the glomerulus, into this capsule fills that capsule with liquid called filtrate. And this is hard to see, but you should look that over in your text. So here is the parietal layer. Forms a continuous layer around the outside. The visceral layer is here. It's got spaces between them and underneath them. The renal tubule is about 3 centimeters, which is about an inch, 1.2 inches long. Consists of a single layer of epithelial cells. Each region of that tubule has its own unique histology and function. And we divide those tubules into 3 major parts: proximal, nephron, and distal. So the proximal convoluted tubule is proximal because it's closest to the renal capsule. The nephron loop goes into the kidney. It can be a long loop or short loop. We'll get that later. And the distal convoluted tubule is twisted, and it's further from the renal corpuscle. That's why we call it distal. The distal convoluted tubule is going to drain into those collecting ducts we spoke of earlier. So the proximal convoluted tubule, you can get from the name, it's close to the renal capsule. It's quite twisty, it's convoluted, and it's a tubule. So those cells are cuboidal cells. They have dense microvilli that form a brush border. That increases their surface area. They also have a lot of mitochondria because there's going to be a lot of active transport going on in these cells, so they need ATP for energy. They are functioning in reabsorbing and secreting materials into the filtrate, and they are entirely within the cortex. Here are convoluted tubule cells. The nephron loop was traditionally referred to as the loop of Henle, again after the anatomist who described it, but nephron loop is a more descriptive term. The U-shaped structure has 2 limbs, one that goes down and one that comes up. So we start with the descending limb, which is the proximal part of the loop. And the proximal part of that loop, the descending limb, is continuous with the proximal tubule. The distal portion is also called the descending thin limb. It is simple squamous epithelium. And then going back up, we have the ascending limb, which is also called the thick ascending limb. It is thin in some nephrons. We'll get to those details later. But the cells that make it up are cuboidal or columnar cells. The distal convoluted tubule, further away from the renal capsule, the other side of the nephron loop, but still convoluted, twisty, has cuboidal cells, very few microvilli. It is involved more in secretion than reabsorption, and that is, like the proximal convoluted tubule, confined to the cortex. Here's a renal cortex section, and you can see this glomerulus. You have the capsule and the capillaries inside. Collecting ducts. Within the collecting ducts, there are 2 cell types: principal cells and intercalated cells. The principal cells are sparse. They have short microvilli. They are very important in maintaining water and sodium balance. The intercalated cells are cuboidal cells. They have a lot of microvilli, and they come in 2 different types, A and B. They are both involved in maintaining acid-base balance of the blood. The collecting ducts receive filtrate from many nephrons. They run through the medullary pyramids and give the pyramids their striped appearance. Those ducts fuse together to deliver urine through the papillary into the minor calyces. And again, I encourage you to look through this image in your book, but we have our glomerulus with its capsule around the outside, the capillary tuft inside. Filtrate is going to fill that capsule and make its way through the proximal convoluted tubule into the nephron loop, back up into the distal convoluted tubule, and finally into collecting ducts. There are two major groups of nephrons. They are either cortical nephrons or juxtamedullary nephrons. Most of them are cortical nephrons. They make up about 85% of the nephrons, and they are almost entirely within the cortex, including that loop of Henle. The juxtamedullary nephrons are long nephron loops that have the nephron loop of Henle that deeply invades the medulla. The ascending limbs have a thick and thin segment, and those juxtamedullary nephrons are going to be very important when we want to make concentrated urine. So here is a cortical nephron, again almost entirely within the cortex, and a juxtamedullary nephron. The renal tubules are associated with two capillary beds, the glomerulus and the peritubular capillaries. Juxtamedullary nephrons are associated with vasa recta. So the glomerulus is the capillaries that are specialized for filtration. Different from other capillary beds, they are fed and drained by an arteriole. An afferent arteriole enters the glomerulus, and it leaves the glomerulus through the efferent arteriole. So afferent arteriole arises from cortical radiate arteries, and the efferent feeds into either peritubular capillaries or the vasa recta. Blood pressure within the glomerulus is going to be high because the afferent arterioles are larger in diameter than the efferent arterioles, so that makes these arterioles a high resistance vessel. Peritubular capillaries, on the other side of this, are low pressure porous capillaries that are adapted for the absorption of water and solutes. They arise from the efferent arteriole. They cling to adjacent renal tubules in the cortex, and they will empty into venules. The vasa recta are long, thin-walled vessels parallel to the long nephron loops of the juxtamedullary nephron. They arise from the efferent arterioles as well, but they serve the juxtamedullary nephrons instead of peritubular capillaries, and their function is going to be in the formation of concentrated urine. The juxtamedullary complex, or the JGC, involves the modified portion of the distal portion of the ascending limb of the nephron loop. So distal, away from the glomerulus, ascending, going up, and we're talking about the nephron loop, and the afferent arteriole. Important for regulating filtrate formation and blood pressure. Three classes of cells in the JGC: macula densa, granular cells, and extraglomerular mesangial cells. The macula densa are tall, closely packed cells of the ascending limb. They have chemoreceptors, and they are going to sense levels of salt, sodium chloride, in the filtrate. The granular cells, or JG cells, juxtaglomerular, are large, smooth muscle cells of the arteriole. They're going to act as mechanoreceptors to sense blood pressure changes in the afferent arteriole. They are the cells that contain secretory granules with the enzyme renin in them. Remember, renin is important for regulating blood pressure. The extraglomerular mesangial cells are located between the arteriole and the tubular cells. They're interconnected with gap junctions. They pass signals between the macula densa and the granular cells. So here is the JGC complex of a neuron mapped out, and again, I would encourage you to look it over in your text. We're going to stop there, and then the next set will start with the physiology of the kidney.